Hello everyone. Welcome to the new session of Respiration in Plants Part 2. So in this session we are going to study about the anaerobic respiration. In the previous session we have completed about the difference between the aerobic respiration and anaerobic respiration. We also talked about the glycolysis process which occur in you can say all the types of whether it is a prokaryotic cell or a eukaryotic cell or the organism that is following the aerobic respiration and anaerobic respiration. That is the common step we see in the case of both the type of respiration. That is the glycolysis process which occur in the cytoplasm. Okay. Now, next step is uh, the substrate, that is the last end product of the glycolysis, that is the pyruvic acid, that get enter inside the aerobic respiration when the oxygen is available. And the organism that follows such type of respiration is called as aerobic respiration. Okay, so let us see that. For aerobic respiration to take place within the mitochondria, the final product of glycolysis, that is the pyruvate, is transported from the cytoplasm into the mitochondria. And that is the crucial event in the aerobic respiration, right? So, in the process of aerobic respiration, the very important organelle that is involved in this process is the mitochondria. Okay, and the final product, that is the pyruvate or the pyruvic acid, which has been synthesized in the glycolysis, get entered inside the mitochondria, right? So that has been synthesized where? In the cytoplasm. Pyruvic acid is synthesized in the cytoplasm, but they get entered inside the mitochondria. But not as it is. Like pyruvic acid do not get entered inside the mitochondria. It is get converted into the acetyl coenzyme A. Okay, so the very important reaction takes place at the outer membrane of the mitochondria, that is the decarboxylation of the pyruvic acid. Okay, so the pyruvate or the pyruvic acid get decarboxylated right at that outer membrane, and then it is converted to the acetyl coenzyme. This is a very important reaction that takes place. Okay, and now, there are some crucial events that occur at the aerobic respiration. So, let us see that what is that crucial event. The complete oxidation of pyruvate by the stepwise removal of all the hydrogen leave three molecules of carbon dioxide. So, this, you know that pyruvate is a three carbon compound. Okay, and the complete oxidation of pyruvate, it occur by the stepwise removal of hydrogen. So hydrogen atom is removed from the pyruvate and three molecules of carbon dioxide is liberated out, right? So this is the one of the very important steps that occur in the you can say the decarboxylation of pyruvic acid, as well as further oxidation of the pyruvate. Okay, so let us see the other steps that occur in this particular process. The passing on of the electrons removed as a part of the hydrogen atom to the molecular O2 with the simultaneous synthesis of ATP, right? So the electrons, the passing on of the electron means electrons are passed, or you can say removed as the part of hydrogen atom to the molecular O2 with the simultaneous synthesis of ATP. So these are some of the coupled reaction that is occurring simultaneously. Okay, the electrons are passed from one component to the other. Okay, and it is removed, and also the hydrogen atoms are also removed, right? And when hydrogen atoms are removed, one thing is get synthesized. What is that? ATP, right? So hydrogen atoms to the molecular O2, it reaches to the molecular O2 and there is a synthesis of ATP. So what is interesting to note is that the first process takes place in the matrix of the mitochondria, while the second process is located on the inner mitochondrial membrane of the mitochondria. Okay, so The first process is taking place where? In the mitochondria, I mean the matrix of the mitochondria, the liquid substance that is present inside the mitochondria, and that is called as the matrix of the mitochondria. And the second process takes place where? That is the synthesis of ATP. It takes place at the inner membrane of the mitochondria, right? So pyruvate, which is formed by the glycolytic catabolism of the carbohydrate in the cytosol, in the cytoplasm. Cytosol means cytoplasm. After it enter to the mitochondrial matrix, undergo oxidative decarboxylation by a complex set of reactions, okay, and catalyzed by the pyruvate dehydrogenase. So this is a very, very important step of reaction, okay. So first of all, what happened? The pyruvate, which is the end product of the glycolysis that occur in the cytoplasm, okay, it get catabolized. Catabolized means it get broken down, where when it get enter the mitochondrial matrix, okay, oxidative decarboxylation by the complex set of a reaction that is catalyzed by an enzyme, okay, and this enzyme is called as pyruvate dehydrogenase, very important reaction. Okay, so here you can see the reaction catalyzed by the pyruvic dehydrogenase require participation of several coenzymes. Okay, so there are several coenzymes that is required by this particular reaction. So what are those? You can say the coenzymes, including NAD. So here NAD is required, and also the coenzyme A, right? Magnesium ion is also required. Okay, magnesium ion is also required, and of course this reaction take place in the presence of a very complex enzyme that is called as pyruvate dehydrogenase. So you can see here pyruvic acid, okay, then combined with the coenzyme A. So that this is the coenzyme A, and also NAD molecule. These are the coenzyme A and NAD are the you can say coenzymes. Okay, these are the coenzymes, and in the presence of magnesium ion, this pyruvate dehydrogenase is converted into acetyl coenzyme A, and you can see here now acetyl coenzyme is a 2-carbon compound. Okay, it is a 2-carbon compound. So here you can see there is a release of 1 carbon dioxide. So carbon dioxide is removed here. Okay, and NADH2 is also formed. So this is a very, very important step of reaction that is occurring in the matrix of the mitochondria, right? So again, this reaction is catalyzed by the pyruvic dehydrogenase, and that is required. The participation of several coenzyme includes NAD and coenzyme A, right? NAD and coenzyme A. Now, in the presence of pyruvate dehydrogenase, there is a requirement of one, you can say the elements, that is the magnesium ion for the activity of this particular you can say enzyme, okay, pyruvate dehydrogenase, and magnesium ion is very, very important. And this pyruvic acid that combined with the coenzyme A and NAD, right? NAD is reduced to get NADH2, and here 1 carbon dioxide is removed out, and acetyl coenzyme A is formed. So this is very important step of reaction. Now, during this process, 2 molecules of NADH are produced from the metabolism of 2 molecules of pyruvic acid. Now, this is to understand that the glycolysis is started from the glucose molecule. Okay, glucose molecule 6-carbon compound, glucose molecule 6-carbon compound, and it get converted to pyruvic acid, that is a 3-carbon compound, and from 1 glucose molecule you get 2 molecules of uh, pyruvic acid. Okay, 2 molecules of pyruvic acid. Now this 2 molecules of pyruvic acid then form how many molecules of NADH2? 2 molecules of NADH2, 1 by each. Okay, so 1 pyruvic acid get converted to acetyl coenzyme A, 1 NADH2 is formed, And similarly, the another molecules of this pyruvic acid also get converted to acetyl coenzyme, and then another molecules of NADH2 is formed, right? So during this process, two molecules of NADH2 are produced from the metabolism of the two molecules of pyruvic acid, and that is why it is written here in the bracket, produced from one glucose molecules during the glycolysis. Okay, so in the glycolysis process, two molecules of pyruvic acid are formed, right? The acetyl coenzyme then enters a cyclic pathway. Now, this acetyl coenzyme, which has been produced here, that get enter in the cyclic pathway, and that cyclic pathway is called as tricarboxylic acid cycle. More commonly, it is called as Krebs cycle. So Krebs was the scientist who discovered this, and that is why it's this particular, you can say, reaction, or you can say the cycle is called as Krebs cycle after the scientist Hans Krebs, who first elucidated it, right? So this is a very, very important step of reaction. Decarboxylation of pyruvic acid forms the acetyl coenzyme, and acetyl coenzyme is the compound that get enter in the Krebs cycle or tricarboxylic acid cycle. Okay, so this is the starting of the you can say the tricarboxylic acid cycle. So the compound that get enter in the tricarboxylic acid cycle is the acetyl coenzyme A, which is formed from the decarboxylation of pyruvic acid by the enzyme pyruvate dehydrogenase, right? Okay, so let us study about the tricarboxylic acid cycle. The TCA cycle or the tricarboxylic acid cycle start with the condensation of acetyl group with the oxaloacetic acid OAA and the water to yield the citric acid. Okay, so this is very important to understand this tricarboxylic acid cycle. The first compound that get enter in this is the oxaloacetic acid. Okay, now oxaloacetic acid condensed with the acetyl group with oxaloacetic acid. So first compound that accept the acetyl group of acetyl coenzyme A. And you know the acetyl coenzyme has been formed from the decarboxylation of the pyruvic acid, right? So that acetyl coenzyme A combines with the oxaloacetic acid. This is the first step of reaction, oxaloacetic acid, and yield the citric acid. So citric acid is formed, right? And citric acid is the first, you can say, the compound that is stable in this particular reaction. Okay, citric acid. So citrate, or you can say the citric acid, is formed here, and this is occurring in the presence of citrate synthase, the enzyme that is required for the combining the oxaloacetic acid to the acetyl coenzyme A. There is a requirement of enzyme that is called as citrate synthase. Okay, so the reaction is catalyzed by the enzyme citrate synthase. This is a very important step of reaction, and this enzyme is also very, very important. Citrate synthase, and a molecule of coenzyme A is released. So this coenzyme A molecule is now released out. Okay. Citrate is then isomerized to isocitrate. Okay. Now, next step of reaction, what will happen here? The citrate which is formed, okay, in the first step of reaction of this citric acid cycle or the tricarboxylic acid cycle, this is isomerized to isocitrate. Okay, and for this there is a requirement of enzyme that is called as aconitase. Okay, so citrate is converted to isocitrate in the presence of enzyme aconitase. Okay, and it is followed by the two successive steps of decarboxylation. Okay, two times the decarboxylation is occurring here. So, and leading to the formation of alpha ketoglutarate. So, it is followed by the two successive steps of decarboxylation. So, the first decarboxylation reaction, that is the isocitrate, is converted to alpha ketoglutarate. Okay, the isocitrate is first converted to alpha ketoglutarate, and for this there is a requirement of an enzyme that is called as isocitrate dehydrogenase. Okay, isocitrate dehydrogenase, and here again the NADH2 compound is you can say formed. Okay, and one NADH2 molecule is formed, and this is converted to alpha ketoglutarate acid. Now again decarboxylation of alpha ketoglutarate acid is occur, that is the second. It is written here, it is the two steps of decarboxylation. So the first step, isocitrate is converted to alpha ketoglutarate acid, right? And in the second step, this alpha ketoglutarate is then converted to succinyl coenzyme A, and here the enzyme required is alpha ketoglutarate dehydrogenase. Alpha ketoglutarate dehydrogenase, and here again one more NADH2 is formed by the release of carbon dioxide. Okay. So here you can see this is the pyruvate, that is the 3-carbon compound. Acetyl coenzyme A is formed, that is a 2-carbon compound, right? And NADH2 is formed, carbon dioxide is released. Now the 4-carbon compound oxaloacetic acid is combined with the acetyl coenzyme and form the citric acid, that is the 6-carbon compound. Carbon dioxide is released and it is converted to isocitrate. Okay, it is converted to you can say the isocitrate, and then it is converted to alpha ketoglutarate, and by the release of one carbon dioxide, now this alpha ketoglutarate acid is the 5-carbon compound. It is a 5-carbon compound. Okay, now what happened, there is another decarboxylation of the you can say the alpha ketoglutarate acid, and this 5-carbon compound is now converted to succinyl coenzyme A. Okay, succinyl coenzyme A, or then it is converted to succinic acid. So here you can see the carbon dioxide is released out, so 5-carbon compound is converted to 4-carbon compound, and also there is an decarboxylation of pyruvic acid, and there is a formation of GTP. So GTP is equivalent to ATP. So this is the only ATP is synthesized in the citric acid cycle. Okay, only one step at this GTP is formed. Okay, so this you have to keep in mind. Now this is the succinic acid. Succinic acid is converted to another 4-carbon compound, that is the malic acid, and there is an enzyme that is a coupled enzyme, that is isocitrate succinic dehydrogenase. Okay, succinic dehydrogenase, and the malic acid is formed, and then malic acid is later get converted to malate dehydrogenase. Okay, malate in the presence of malate dehydrogenase, it is converted to oxaloacetic acid. So here the conversion of malic acid to oxaloacetic acid, there is a requirement of enzyme that is called as malate dehydrogenase. Okay. So in the remaining steps of citric acid cycle, okay, succinyl coenzyme A is oxidized to oxaloacetic acid. Okay, so in the remaining steps of the citric acid cycle, it is converted to succinyl coenzyme A is oxidized to oxaloacetic acid, and it is allowing to cycle to continue. So during the conversion of succinyl coenzyme A to succinic acid molecule, a GTP is synthesized. Okay, GTP is synthesized. So this is one of the very important step of reaction. Now this is the substrate level of phosphorylation. Okay, substrate level of phosphorylation, and in a coupled reaction, GTP is converted to GDP with a simultaneous synthesis of ATP. I've already told you that later this GTP, okay, is converted to ATP. Also, there are 3 points in the cycle where the NAD is reduced to NADH2, and 1 point there is an FADH2 is reduced to FADH2. If you see the whole reactions, then you can see there is a 3 steps, okay, 3 very important steps that in which this NAD is reduced to NADH2. Okay, NAD is reduced to NADH2, and at one particular point, at only one particular point, that is the conversion of succinate to fumarate. Succinate to fumarate, there is a formation of FADH2 in the presence of enzyme succinic dehydrogenase. Succinic dehydrogenase. This is a very important reaction, and that this particular enzyme is attached to the inner mitochondrial membrane. It is not occurring in the This whole citric acid cycle is occurring in the matrix of the uh, mitochondria, but this particular enzyme is a part of the you can say the inner mitochondrial membrane. It is attached to the inner mitochondrial membrane, and it is also the part of the electron transport system. So when we will study the electron transport system, I'll give the emphasis of this there also, because that is a part of it, right? And in the citric acid cycle, again the succinate dehydrogenase, okay. So this FADH2 is formed. You have to keep in remember on only this one particular FADH2 is formed, whereas the three NADH2 is formed, and one NADH2 is also formed by the conversion of the pyruvate to the acetyl coenzyme A. That is the decarboxylation of pyruvic acid. So the number of NADH2 is formed in the one citric acid cycle, the number of FADH2 is formed in one citric acid cycle, and the number of GTP is formed. You must remember, right? So, the continued oxidation of acetyl coenzyme A via the tricarboxylic acid cycle require continued replenishment of the oxaloacetic acid, the first member of the cycle. Okay, so this is the end of the citric acid cycle, that is the formation of oxaloacetic acid, and this oxaloacetic acid is very important as it only this particular compound that get combined with the incoming acetyl coenzyme, right? Okay. Now, in addition, it also requires the regeneration of NAD, FAD from NADA, FADH2s respectively. And the summary equation for this phase of the respiration may be written as so. Overall, the tricarboxylic acid cycle, you can write it here. So it is pyruvic acid, okay, and it is a 4 NAD. See here, 4 NAD is required and it is converted to 4 NADH2. So total, you are starting from the decarboxylation of pyruvic acid and the carboxylic acid cycle. Okay, so I've already told you decarboxylation of pyruvic acid, one NADH2 molecule is formed as it is converted to this pyruvic acid to acetyl coenzyme A. And later the another NADH2 molecule is formed in the tricarboxylic acid cycle. So total 4 NADH2. Okay, one FADH2 succinate dehydrogenase enzyme. Okay, that is a part of the inner mitochondrial membrane, and that convert the succinate to fumarate. Okay, succinate to fumarate, and here one FADH2 molecule is formed. Okay, and here it is written ATP plus inorganic phosphate. Actually, it is a GTP, but later this GTP is again get converted to ATP molecule. There is no difference in there, and this is occurring in the mitochondrial matrix. Okay, so we have till now seen that the glucose has been broken down to release carbon dioxide and 8 molecules of NADH2. So why it is written here 8 NADH2? Because there are two pyruvic acid is formed from one glucose molecule. So it has been seen that glucose has been broken down to 2 molecules of pyruvic acid, and 1 molecule of pyruvic acid in the tricarboxylic acid cycle is giving rise to 4 NADH2. So 2 pyruvic acid will give rise to 8 NADH2. Okay, 8 NADH2 molecule, and in the same way, 2 FADH2 molecule has been synthesized beside just 2 molecules of 2 ATP in the tricarboxylic acid cycle. So this is the overall, you can say, the conversion of the glucose to the what will be the end product of the tricarboxylic acid cycle. So here we can see that. Now, you may be wondering why we have been discussing the respiration at all. Neither O2 has come into the picture nor progress, large number of ATP has yet been synthesized. So this is a very important thing to understand. Okay, we are studying the aerobic respiration, but here we can see that there is no oxygen has been used till now. Okay, and there is no you can say that ATP has been synthesized, large number, right? So why this Krebs cycle is so important? So also what is role of the NADH2? But here 8 molecules of NADH2 and 2 molecules of FADH2 is formed. So what is the role of that, right? And let us now understand the role of O2 in respiration and how ATP is synthesized, right? So let us understand this. Electron transport system and oxidative phosphorylation. So this is very important, okay, to understand what is the electron transport system and how the oxidative phosphorylation takes place. So the following step in the respiratory process are to release and utilize the energy stored in NADH2 and FADH2. Okay, so these are the steps that are involved in the respiratory process, and it release out energy that is stored. Okay, so this NADH2 and FADH2 is not a waste. Okay, this is the ultimate thing that is going to get converted to ATP molecule via electron transport system. So that is why these NADH2 and FADH2 are very, very important. Now, this is accomplished when they are oxidized through the electron transport system, and electrons are passed on to the O2, resulting in the formation of H2O. Okay, so here this is very important. At the last, what will happen? There is an oxygen molecule that accepts the electron as well as hydrogen atom, and it will form the water molecule. So that is why this oxygen is so very important. Okay, this oxygen is very, very important. So this is accomplished when they oxidize. That is why it is written here, oxidized, and phosphorylation is takes place, and that is why it is called as oxidative phosphorylation. Right, so this is accomplished when they are oxidized through the electron transport system. So there is a chain of you can say the step of reaction in which the electrons are transported from one compound to the another, and there is a release of electron, and that ultimate it is passed to, at the end it is passed to the O2, where it get combined with the hydrogen molecule and it forms a water molecule. Okay, the metabolic pathway through which the electron passes from one carrier to the another. It is called as electron transport system. Very important. Okay. So this is one of the metabolic pathway. Okay, in which the electrons are passes from one carrier protein to the another, and this is called as electron transport system, and it is present in the inner mitochondrial membrane. So it is a part of the inner mitochondrial membrane, right? So here you can see the electron transport system. It is having the first component here. So basically it is having the 4 component, right? So here you can see the complex 1, complex 3, then here complex 2, okay, and then here complex 4. And there is also the fifth component, okay, that is the ATP synthase, okay, ATP synthase. So 4 components are there, right? So here you can see this is the inner membrane space. There must be an outer membrane. Then there is this is the inner membrane, and in between the outer membrane and inner membrane, there is a intermembrane space. Okay, the space that is present in between the outer and inner membrane. Okay, now this is the inner membrane, and to the inner membrane, there is a matrix of the mitochondria, right? So, okay. So this is the complex one, NADH dehydrogenase. So you must remember the names of these complexes. Okay. So these complexes are the part of the this part of the inner mitochondrial membrane. They are present in the intrinsic protein form. So this complex one, that is NADH dehydrogenase. Okay, so it is having the iron sulfur protein and FMN molecule, and this passes electron to the ubiquinone, and this ubiquinone lately forms the transport to the third It is transport to the third complex that is called as cytochrome BC1 complex. Okay, cytochrome BC1 complex. So here you can see the cytochrome B is there, then iron sulfur protein, then cytochrome C1, and that is why it is written known as cytochrome BC1 complex, complex 3, right? Okay. Now, there is a complex 2. Complex 2 is known as succinate dehydrogenase. Now, I've already told you at the time of succinate dehydrogenase, where this succinate which has been synthesized in the tricarboxylic acid cycle is converted to fumarate in the presence of succinate dehydrogenase. So this was the enzyme succinate dehydrogenase. Okay, so FADH2 is synthesized in the presence of this particular enzyme. And later the fourth complex. So here you can see the fourth complex, copper and cytochrome A containing, you can say the complex. Okay, CuA, then cytochrome A, cytochrome A3, then CuB. Okay, this is the sequence of the copper and the cytochrome A. CuA, cytochrome A, cytochrome B, and then CuB. Okay, so this is the cytochrome 4 complex, which is also known as cytochrome C oxidase. Cytochrome C oxidase. Okay, so let us start here. The NADH2, which has been synthesized in the decarboxylation of pyruvic acid, okay, and also the 3 other steps in the citric acid cycle in which NADH2 has been synthesized. So comes here. All the NADH2 which has been synthesized, whether it is a glycolysis process, whether it is a decarboxylation of a pyruvic acid, or the further decarboxylation process in the tricarboxylic acid cycle. So all the NADH2 has been synthesized there, comes here. Okay, and it transports the electron. Here you can see it is transporting the electron to the complex third. From complex third it reaches to the cytochrome C, and from cytochrome C it is reaches to the fourth compound, that is the cytochrome C oxidase. Now here you can see this, then how this it is linked with the complex 2. So complex 2, if you remember, the succinate, this succinic acid or the succinate has been converted to fumarate. So cytochrome succinate dehydrogenase, and the FADH2 is formed. So here you can see it is transporting the FAD. Okay, it is transporting the FAD to ubiquinone QH, and then it is transporting to the cytochrome C. So this succinate to fumarate, it is get converted in the presence of this, and the hydrogen is transported here. Okay, so ubiquinone, here you can see it is transported to in the form of UQH2, and then electron from here it is transported to cytochrome B, then it is FES, cytochrome C1, then cytochrome C comes out from the complex 3, and then it is transported to complex 4, right? So FADH2, this is the conversion of FADH2. Okay, now what happened if you see here the matrix in the conversion of NADH to NAD. Okay, the hydrogen is transported to the inner membrane space, intermembrane space, and here also the complex 3. Okay, it is also again transported the hydrogen here. From FADH2, again the hydrogen is transported here. Okay, and here also you see there is a conformation of the water molecule. Okay, and then again 2 hydrogen atom is transported here. So the concentration of hydrogen is much high in the intermembrane space. Okay, in the space between the outer membrane and the inner membrane. So this hydrogen is transported later. The hydrogen is transported from this ATP synthase, okay, to the inner mitochondrial membrane, and ATP is synthesized, right? So electron from the NADH produced in the mitochondrial matrix during the citric acid cycle are oxidized by an NADH dehydrogenase complex 1, and electrons are transferred to the ubiquinone located within the inner membrane, right? So NADH dehydrogen, that is the first component of the you can say the electron transport system, complex 1, and then it is transported the electrons. Electrons are there. From here, it is transported to the ubiquinone, that is located to in the inner membrane. Now, ubiquinone also receives reduced equivalent via FADH2, that is the complex 2, and it is generated during the oxidation of succinate to citric acid cycle. The reduced ubiquinone or ubiquinol is then oxidized with the transfer of electron to the cytochrome C via cytochrome BC1 complex, that is the third complex. Okay. So whatever the ubiquinol that has been reduced is then oxidized. How it is oxidized? By transferring the electron to the next. Okay. With the transfer of electron to the cytochrome C. So here ubiquinone is transported its electron to cytochrome C via BC1 complex, that is the complex 3. Okay. Cytochrome C is a small protein attached to the outer surface of the inner membrane and acts as a mobile carrier for transfer of electrons between the complex 3 and complex 4. So this is the one of the cytochrome that makes you the connection between this complex 3 and complex 4. Okay. So cytochrome C, it is a very small protein that is attached to the outer surface of the inner membrane. Okay, outer surface of the inner membrane and acts as a mobile carrier. And it is acting as a mobile carrier for the transportation of electrons between the complex 3 and complex 4. Complex 4 refers to the cytochrome C oxidase complex containing cytochrome A, A3 and the 2 copper center. Okay. So complex 4, it refers to cytochrome C oxidase. Okay. Fourth one is known as cytochrome C oxidase. And it is containing, okay, it is containing the 4 things: cytochrome A, A3 and 2 copper center. Okay. It is having the 2 copper center. So when the electron passes from one carrier to the another via complex 1 to complex 4, through the electron transport chain, they coupled to ATP synthesis. Okay. This electron, when it is transported, it is transferring the hydrogen ion from matrix region to the inner intermembrane space. Okay, this transportation of electron is coupled with that. And at the last, you see there is a formation of ATP molecule. So how the ATP molecule was formed? Because of this large hydrogen ion concentration that is developed via transportation of electron in the intermembrane space. Okay, so that is why it is written here. They are coupled to ATP synthase, that is the fifth complex for production of ATP from ADP and inorganic phosphate. So the number of ATP molecules synthesized, it depends on the nature of electron donor. Okay, so how many number of ATP molecules are synthesized? It is depending upon the nature of the electron donor. Oxidation of one molecule of NADH give rise to 3 molecules of ATP. So here you can see oxidation of one molecule of NADH. Okay, one molecule of NADH, it is forming 3 molecules of ATP, while that of the 1 molecules of FADH2 that produces 2 molecules of ATP. Only 2 molecules of ATP are synthesized, right? So although the aerobic process of respiration take place only in the presence of oxygen, the role of oxygen is limited to the terminal stage of the process. So you can see here at the terminal position, of the you can say this electron transport system, there is a requirement of oxygen at the terminal position. Okay, so we can say the rest of the reaction can take place. No, the rest of the reaction will only take place when there is a presence of oxygen molecule at the terminal position, right? So although the aerobic process of respiration take place only in the presence of oxygen, the role of oxygen is limited at the terminal stage of the process. Okay. Yet the presence of oxygen is vital. Okay, so this presence of oxygen is very vital since it drives the whole process removing of hydrogen. So you can see this. Electron is moving, hydrogen is transported from the matrix region to the intermembrane space. Okay, so this is occurring because of the presence of oxygen at the terminal position. If the oxygen is not present at the terminal position, then this reaction will not take place. Okay, right? So oxygen act as a final hydrogen acceptor. It is acting as a final hydrogen acceptor. Unlike photophosphorylation, where it is a light energy, okay, light energy that is utilized for the production of proton gradient required for the phosphorylation. In respiration, it is the energy of oxidation reduction utilized for the same process. So if you compare with the, that is the photosynthesis, okay, photophosphorylation, that is the light reaction, photophosphorylation. Okay, so if you compare with that, what what is the driving force for the synthesis of ATP? There, the photophosphorylation, where the light energy is utilized for the production of the proton gradient. There, the proton gradient is responsible for the synthesis of the ATP, right? But here, in the respiration process, the energy, it is the energy of oxidation reduction that is utilized the same process, means the production of ATP. So it is for this reason that the process is called as oxidative phosphorylation. So that is why this whole process is called as oxidative phosphorylation, that is occurring in the presence of oxygen, and it is a oxidation reduction based reaction. The transportation of electron here, that is the oxidation and reduction based reaction. It is not like you can say the driving force is not the light, as we have seen in the case of a photophosphorylation. So here you can see inner mitochondrial membrane, and inner mitochondrial membrane, there is an F1 particle, or you can say the ATP synthase enzyme. And ATP synthase enzyme is in the form of like you can see the F0 part and F1 part. So F0 part is embedded in the membrane, and F1 particle is like here, right? And you know that the intermembrane, this is the inner mitochondrial membrane, and there must be an outer mitochondrial membrane, and in between there is a intermembrane space where the hydrogen concentration is very high, and that hydrogen concentration has been reached to so high because of the oxidation reduction reaction that is taking place via the transportation of the electrons, okay, from one complex to the another, okay, and this transportation of electron from one complex to other is coupled with the transportation of hydrogen from the matrix region to the inner mitochondrial membrane space. Okay, inner mitochondrial membrane, intermitochondrial membrane space. So here you can see now the two hydrogen ion is transported back to the matrix region, and this ADP, okay, and Pi get uh, coupled to in the F1 particle, and ATP is synthesized. So every two hydrogen molecule when get enter inside this matrix region, okay, ATP synthase will synthesize one ATP molecule. So this is the diagrammatic presentation of ATP synthesis in the mitochondria. So now you can understand how this hydrogen ion is so important, because this is the ultimate, you can say the result of the conversion of NADH2 and FADH2 molecules in the form of ATP, right? And that is why we say that one NADH2 molecule will synthesize 3 ATP, whereas one FADH2 will synthesize only 2 molecules of ATP. The F1 headpiece is a peripheral membrane protein complex and contain the site of synthesis of ATP from ADP and inorganic phosphate. So this ATP synthase is having the two part. F0, which is you can say the embedded in the membrane. Okay, it is a membrane protein, and F1 is the protruded out, and it is present in the matrix region. Okay, so F1 headpiece is more important. It is a peripheral membrane protein complex, and it contains the site of synthesis of ATP. F0 is an integral membrane protein complex that forms the channel through which the proton cross the inner membrane. Okay, so this is the channel through which the F0. Okay, F0 is making a channel. Okay, it is having a channel through which hydrogen molecule is transported from the inner membrane space, intermembrane space, towards the F1 particle, F1 part of this ATP synthase. Okay, so F0 is integral membrane protein complex that forms the channel through which proton crosses the inner membrane. Now, the passage of proton through the channel is coupled to the catalytic site of the F1 component of the production of ATP. Okay, so this is the passage of proton. Now, what will happen if the proton is passed? Then the passage of proton through the channel is coupled. Okay, it is joined, or you can say the coupled to the catalytic site of the F1 component of the production of ATP. So unless there is a transference of the hydrogen molecule from the intermembrane space of the mitochondrial membrane, okay, there will be no synthesis of ATP. So it is very, very important step. For each ATP produced, 4 hydrogen access through F0 from the intermembrane space to the mitochondrial down to the electrochemical proton gradient. Okay, so 4 hydrogen must pass through the F0 from the intermembrane space to the matrix region down to the electrochemical proton gradient. Okay, and the ATP is synthesized. It is possible to make the calculation of the net gain of ATP for every glucose molecule oxidized. So you can calculate, okay? You can calculate how much glucose molecule, one glucose molecule can give how much amount of the ATP, how much ATP is formed. But it is reality, this can remain only theoretical exercise. So these calculation can be made only on the certain assumptions that there is a sequential orderly pathway functioning. So whatever we have studied, okay, the sequence of the reaction, that is only occurring. It is not going from here and there, because the every steps you see the product that is formed, it is coupled with some other reaction also. We will study the amphibolic pathway, then you will understand. But we assume at that one particular time, this whole step of reaction is taking place as it is. Okay, so there is a sequential orderly pathway functioning with one substrate forming the next, and with the glycolysis tricarboxylic acid cycle electron transport system pathway following one after the other. So this is the sequence of reaction that is taking place. Okay, the NADH synthesized in the glycolysis is transferred into the mitochondria and undergoes oxidative phosphorylation. So NADH2, which has been synthesized in the glycolysis process, it is transferred inside the mitochondria, and that undergo oxidative phosphorylation process. None of the intermediate in the pathway are utilized to synthesize any other compound. Okay, we assume that there is no intermediate compound is coupled with the product that has been formed in these steps of reactions. Okay, so that is why it is mentioned here. None of the intermediates in the pathway are utilized to synthesize any other compound. No compound has been synthesized. One glucose is being respired on other alternative substrates are entering the pathway at any of the intermediary stages. So, but this kind of assumptions are not really valid in a living system. It is not occurring in the reactions, the stepwise reaction that we have seen in the living system. All the pathway work simultaneously and do not take place one after the other. So there are so many reactions that is taking place inside the cell. Okay, so substrate enters the pathway and are withdrawn from it, and when necessary, ATP is utilized as and when needed. So enzymatic rates are controlled by the multiple means. So there are so many reactions that are coupled with one of the reactions. This is the part of that is the part of the anaerobic respiration. So it is not the same way which we have studied. Okay, so yet it is useful to do this exercise to appreciate the beauty of the efficiency of the living system in extraction and storing energy. But it is important to understand that if the reaction is taking place in the stepwise, then how much ATP is synthesized. So hence there can be net gain of 38 ATP molecules. Okay. So at the last we can say that 38 ATP molecules are synthesized during the aerobic respiration of 1 molecules of glucose. Okay. If we use 1 glucose molecule, then 38 ATP molecule has been synthesized. So now let us compare the fermentation and aerobic respiration. Okay. So we have seen the fermentation process, that is the anaerobic process, and we can compare with the aerobic respiration. Fermentation account only a partial breakdown of glucose, whereas the aerobic respiration is completely oxidized. The food is completely oxidized or completely degraded, and it has been changed to carbon dioxide and water. This is the end product, complete oxidation of glucose. Okay. Now, in the fermentation, there is a net gain of only 2 molecules of ATP. Only 2 molecules of ATP for each molecule of glucose degraded to pyruvic acid, whereas many more molecules of ATP are generated under the aerobic condition. So, you can compare how efficient this aerobic respiration is. NADH2 is oxidized to NAD rather slowly in fermentation. However, the reaction is very vigorous in the case of aerobic respiration, right? In the case of aerobic respiration also, the NADH2 which has been formed is converted to ATP in a very fast way. Whereas in the case of NADH2 which has been synthesized in the fermentation process, is changed to NAD in a very slow process, very slow. Now, glucose is favored substrate for the respiration. Okay, this is one of the favored substrate for the respiration, and all carbohydrates are usually first converted into glucose before they used for the respiration. So if there is a carbohydrate, protein, fat, okay, so first preference will be given to the carbohydrate, especially the glucose. Other substrate can also be respired, as has been mentioned earlier, but then they do not enter the respiratory pathway at the first step that we have started with the glucose. It will not be like that. Okay. So we can see the figure and we can understand how the respiratory pathway works out. So here you can see carbohydrate. It is converted to the simple sugar, that is the glucose. Then glucose is converted to the glucose 6-phosphate, fructose 1,6-phosphate. We have seen this in the case of a glycolysis. Okay. Then dihydroxyacetone phosphate, pyruvic acid. Then there goes to the acetyl coenzyme and then get it entered to the Krebs cycle. And later it is converted to goes to the electron transport system. Right. So this is the step which we have studied. Now you can see the proteins. The proteins are converted to amino acid, and this amino acid can also get converted to the pyruvic acid. So it is skipping all that glycolysis process, right? Now, if you see the fats, fatty acid and glycerol. Now, this fatty acid, it is broken down. Fats is broken down into the fatty acids and glycerol, and this fatty acid can get entered here. It is changed to acetyl coenzyme and it can get entered in the Krebs cycle directly. So it is skipping all that glycolysis process. But what about this glycerol? This glycerol can change to the dihydroxyacetone phosphate and it can get entered in the glycolysis process at this particular step. And it is followed the further steps as we have seen in the case of glucose molecule, right? So this is the amphibolic pathway, right? So fats would need to be broken down into the glycerol and fatty acid first, and if fatty acid were to be respired, they would first be degraded as an acetyl coenzyme A and enter the pathway. Glycerol would enter the pathway after being converted to phosphoglyceraldehyde. The protein would be degraded by the proteases and the individual amino acid, that is the deamination process. Okay, deamination of amino acids, and it is converted to the pyruvic acid. Okay, depending on their structure, would enters the pathway at the same stage within the Krebs cycle or even the pyruvate or acetyl coenzyme. So since the respiration involves breakdown of substrate, the respiratory process has traditionally been considered as a catabolic process. It is a catabolic process, and the respiratory pathway as a catabolic pathway. But it's this understanding correct? We have discussed above at which point the respiratory pathway different substrate would enter if they were to be respired and used to derive energy. So what is important to recognize that it is these very compound that would be withdrawn from the respiratory pathway and synthesis of the set substrate. Okay. So fatty acid would be broken down to acetyl coenzyme A before entering the respiratory pathway. It is used as a substrate. So acetyl coenzyme A can be used as a for the different reaction. In many of the reaction, it is used as a substrate, right? But when the organism need to synthesize fatty acid, when there is a requirement of this fatty acid, this acetyl coenzyme would be withdrawn from the respiratory pathway of it, and hence the respiratory pathway comes into the picture both during the breakdown and the synthesis of fatty acid pathway. Okay, so sometimes what happens, there is a requirement of the synthesis acid pathway. So this acetyl coenzyme A get converted to fatty acid. Whatever the acetyl coenzyme A, sometimes it is converted from it is broken down from the glucose to acetyl coenzyme A, and this acetyl coenzyme A moves to the synthesis of the uh, fatty acid, synthesis of the fats. Okay, and at that particular time, you can see the breaking down of this glucose molecule has been ceased away. Similarly, during the breakdown of the synthesis of protein 2, the respiratory intermediates form the link. Breaking down process within the living organism is the catabolism, and synthesis is the anabolism. Or because the respiratory pathway is involved in the both the anabolism and catabolism, it would be better to consider the respiratory pathway as an amphibolic pathway. So we cannot only just say this respiratory pathway is just the catabolic pathway. It is actually the anabolic pathway, because some of the substrate can get enter in the synthesis of amino acid, and some can get enter in the process of fatty acid synthesis as well. Okay, now we will study about the respiratory quotient. Okay, and what is the understanding of this? So let us now look at the another aspect of the respiration. As you know, that during the aerobic respiration, O2 is consumed and CO2 is released. So we all know that O2 is consumed in the respiratory pathway and CO2 is released out. Now the ratio of the volume of CO2 evolved to the volume of O2 consumed in the respiration is called the respiratory quotient or the respiratory ratio. So respiratory ratio or quotient can be understand that is the volume of CO2 evolved in the reaction. Okay, how much carbon dioxide is evolved is divided by the volume of O2 consumed. How much O2 is consumed. Okay, so this will give the RQ value. The respiratory quotient depend upon the type of respiratory substrate used during the respiration. Okay, so it is very different in the different types of you can say the substrate that has been used in the process of respiration. Now, when the carbohydrates are used as a substrate and are completely oxidized, when the carbohydrate is completely oxidized, the RQ will be 1. At that time, RQ will be 1. And because equal amount of carbon dioxide and oxygen are evolved and consumed, respectively as shown in the equation below. So here you can see C6H12O6 plus 6O2 gives 6CO2 plus 6H2O plus energy. That is the complete oxidation of glucose molecule. So here you can see 6 molecules of carbon dioxide has been produced and 6 molecules of oxygen is being used. So if you divide, then you will get 1, right? So the RQ value for the carbohydrate, it is 1. Now, when fats are used in the respiration, then the RQ is less than 1. Okay, so calculation for the fatty acid, that is the tripalmitin, it is used in the substrate is shown here. So tripalmitin, here you can see 145 O2 is used and 102 carbon dioxide is produced in this oxidation of the tripalmitin. So RQ value will be 102 carbon dioxide divided by 145 O2 used. So you will get 0.7. When protein are the respiratory substrate, then the ratio will be 0.9. So what is important to recognize is that in living organisms, respiratory substrates are often more than 1. It is not always carbohydrate. And the pure protein or fats are never used as a respiratory substrates. Okay. So this is all about this session. We will meet with another session. Till then, take care and bye.